I want to thank everybody for attending the short webinar today. So my name is Ryan Donnelly, Product Manager for the Gene Editing Portfolio within Horizon Discovery. And I just wanted to, the topic for this presentation will be around tips for a successful gene editing experiment. So this will be anywhere from folks that have tried some editing experiments in their lab and maybe they didn't work out well, or for folks that know they need to bring editing into the lab but are kind of confused on where to start. This will go through kind of some key considerations from for all the steps across the process. And for those of you unfamiliar with Horizon Discovery, I just want to take a quick moment to kind of explain who we are. So Horizon's goal is really to empower scientists wherever their gene editing experiments take them. We have a load of different types of reagents for whatever type of application, whatever downstream analysis, whichever cell type that you're editing in. We'll likely have a reagent format that will work for what you're trying to do. So for folks that have looked into gene editing a bit, there's two key reagents that are necessary. You'll need a Cas9 nuclease as well as a guide RNA. For the guide RNA side of things, we have pre-designed knockout guides for genome-wide coverage for human, mouse, and rat genomes. So if you have a gene within any of those genomes that you want to knock out, feel free to check those out. They're available in both synthetic as well as lentiviral formats, and we can get into that a little bit later of how you can select one over the other. And then if you're working in organisms outside of human, mouse, or rat, maybe you're working in a non-standard editing nuclease, or if you need location-specific editing, in the case of doing knock-in experiments where you're editing a specific region within your transcript to knock in a donor template, we have a design tool that can help with that. So yeah, outside of the guides, I mentioned that you'll need a Cas9 nuclease. So similar to our guides, many different types of formats available that we can match up based on the cell type and what guide format that you're using. We can get into a little bit of what that looks like later on in the presentation. And the nice thing is the reagents can scale anywhere from an individual gene, even an individual guide RNA. We have multiple designs for each gene. Those can scale up to predefined libraries as large as whole genome libraries or subsets of genomes. And that's really where I'm going to leave the product side behind. This is not a product-based webinar. We really just want to get these tips and tricks out there for folks that are starting their gene editing experiments. And one other thing I'll note is we won't touch on how CRISPR works. There's many, many better webinars out there, so I won't attempt to take that on. If that's a question, there's many sources where you can find that. But with that, we'll jump into kind of what are these key considerations that are necessary when you're planning, executing, and analyzing your gene editing experiment. So when I was thinking about this webinar, the key question came up in my mind was, how would we define success of a gene editing experiment? And to me, when I think of that, it's that the data that we generate answers the hypothesis set forward. So if we define success as that, what are some key metrics that we want to be able to check off so we can have success? And the first one that came to mind was we want the data generated in the required cell type of interest. If we're asking questions around, well, what would happen if I knocked out this protein in a primary immune cell, we would want to edit in a primary immune cell line instead of an immortalized cancer line, which can be more predictable. Editing in primary cells can be difficult, though. So we want the data to be as closely aligned with that hypothesis that we can. We want high-quality data so that the analysis is straightforward on the back end. We don't want to have to screen thousands of clones if we can avoid it. Sometimes we can't, but we want the analysis on the back end to be as straightforward as possible. And then the last thing that came up was we want our downstream analysis, we want the results of that to answer that question. So if we're doing a knockout experiment, we're likely going to be looking at phenotypic change. Compared to if we're doing a knock-in experiment, we may just want to know the sequence of that edited cell line. So based on that criteria, the way in which we analyze our cells at the end will be different. So maybe we'll sequence for knock-ins, and maybe we'll look at a phenotypic assay for our knockout questions. And we'll get into a little more details around all of these as the presentation progresses. But the box that I have here kind of on halfway down the page is what we've thought through as what are the key criteria, what are the critical checkpoints that we pass through when we're designing a gene editing experiment to when we actually are conducting the wet bench experiment, and then when we're analyzing the results. What's necessary for us to have success? And we've broken it out into a few different areas. So before our experiment, as we're doing the planning, we'll look at cell line selection, the impact of the mutation of interest, how to go about designing guide RNA, and then which format to select for our reagents. So for our Cas9 nuclease as well as our guide RNA, which format of those makes the most sense for our cell line? And then we move into doing the experiment itself, doing the reagent delivery optimization. So there's a few different ways to get our reagents into the cells. Let's make sure we're doing that to the highest efficiency that we can so we can have high editing efficiency on the back end. And then enriching for edited cells. This can help with allowing for fewer clones to be followed up with when we actually do the analysis. So we'll walk through each of these examples in a little more detail. So to start, the first thing that we'll want to look at is we want to make sure we're using a well-characterized cell line. And there's a few different reasons for that. But what the cell line should look like, in principle, the first thing is it should have low copy numbers, and for both knock-in and knock-out experiments. And the reason for that is if we have polyploidy within our cell line, it's going to be much more difficult to get homozygous knock-in clones or homozygous knockouts to really create a true functional protein knockout. It's just there's more copies of that gene to edit. So if we can get by with using a diploid line, that's likely where we would want to be. The next is the cell line should have well-characterized transfection protocols. So this can help with, we need to get our reagents into the cell to do editing. If we don't do that, we're going to have very poor editing. So having cell lines that have well-characterized protocols, very efficient protocols, this can help with getting those reagents in and the downstream success of high editing efficiency within our cells. And lastly, we want the cell lines to be very well annotated. We want to know where the SNPs are when we go about designing our guide RNAs, because that can actually have an effect on editing efficiency as well. If we have unknown variants, because just the cell line's poorly annotated, and we design guides near that region, our guides may not sit down well and edit because of that. So there's a theme emerging here. We want high editing efficiency regardless of the application. And kind of the steps that we'll take along the way through this webinar will make sure that we're aiming to achieve the highest level of efficiency possible. There's a couple unique characteristics we should look for within a cell line if we're going to try to create a clonal line, so grow up from a single cell. We can imagine, if we're trying to grow up from a single cell, that we need cells that can be grown up from a single cell. There are some cell lines that do not grow up well if they don't have neighboring cells to grow up with. There are some ways around this. We can do low concentration kind of plating to where maybe it's not one cell per well, but maybe a handful, and then do sequential dilutions and growing up of those cells to dilute out the different cells that exist to eventually get to a clonal line. So there are ways around it, but it's not ideal. It takes a long time. Another way to reduce the time is just have cells that divide rapidly, and that'll shorten the clonal isolation times. The next thing that we'll want to be aware of is kind of twofold. So we need to know what the type of, if we were to mutate the cell line of interest with our specific edit, what is that going to do to the cell line? And then next, how do we go about designing a functional guide RNA? So the first thing, if we look at the desired mutation aspect, if we were to mutate our cell line with the specific change that we want, is that going to impact the viability of cell growth? If so, that will likely lengthen the downstream analysis times, just because for all of these types of experiments, we need to grow up the cells to analyze them on the back end. So just to be something, just to be aware. And then a more interesting one is if we were to target an essential gene, so a gene that if it does not exist, the cell dies, and we look to knock it out, that likely won't work well. And the reason for that is if we knock out all the copies of the functional gene, the cell will die, so there won't be anything left to analyze. So likely what will happen is if you try to do that, you can get heterozygous mutation, so you may knock out one copy of the diploid genome, or a couple copies of a polyploid genome. Or you can get a truncated yet still functional protein. So these are all just things to, to keep in the back of your mind as you're setting up the experiment. The next thing to look at will be how do we go about designing a functional guide RNA? So there's a few things that we should be cognizant of. The first is that we want to design our guides against the most relevant isoforms. So this is important so we don't get truncated proteins or different cleavage patterns. The next goes back to the last slide of we want to make sure that we're editing in regions where SNPs don't exist, if at all possible. And the reason for that, again, is we want to make sure that our guide RNA will bind as tightly as possible to the targeting region. If there's SNPs there, we may not have full binding, and that can have that can lower our editing efficiency. And then the last bullet here is we want to make sure that the guides themselves are highly specific. So while we want guides to edit our region of interest, and we want them to edit at a very high efficiency, we don't want them to edit other areas of the genome that may have high homology, such as pseudogenes or other areas that have similar sequence patterns. And there's a few ways to do that. You can run specificity checks, but it's critical that we make sure that the guides are not or are limited in the amount of off-target alignments that they target. So when designing guide RNAs, there's a few options. You can use online tools to design them yourself, or if you want a company to do it for you, Horizon does offer pre-designed guides for knockout. So this is the left side here. All you need to do, and this is for human, mouse, and rat, and genome-wide, you'll enter your gene of interest that you want to knock out, and multiple guides that target that gene will show up. And then you can just select however many guides that you want to test out for your experiment. And these are available as synthetic as well as lentiviral backbones, and then those can be packaged into particles if necessary. If you're looking to do knock-in work, or you're working with a species outside of human, mouse, or rat, or you're using a nuclease that's different than S-pyogenase Cas9, that is the traditional gene editing nuclease, there are ways around that too. So we have a design tool that you'll enter in your organism, and right now we can design for 40 different organisms. Type in your gene of interest, and then you'll select from the drop-down menu your nuclease. So you may be able to see in there, we have S. pyogenes Cas9, Staph aureus Cas9, and MAD7. Those are all preloaded with their specific PAMs, repeat sequences, how long the target needs to be. But then there's an other option there too. So if you're working with an editing nuclease that is not one of these three, easily select other. Those grayed out boxes will become white, and then you'll just enter in the specific information for that nuclease. So you'll have to enter in the PAM, repeat sequence, how long the target is, based on that nuclease, and then you can design for that. And what ends up happening when we have the organism, the gene, the nuclease, we'll hit next, and then the transcript for that region will show up. What you'll do here is move the cursor around to where you want to edit that gene of interest. So this is very important for knock-in experiments because we want to edit a specific region of that transcript. So for this example, I move the cursor right after the start codon and hit enter, and then the guides start showing up. The guide designs that will work with this specific nuclease that was selected start showing up. And you'll see a couple different metrics there. The first is a specificity call, low, medium, and high. Ideally, you want high specificity, as we talked about, low off-target effects if we have that. And there's a proximity metric. So this relates to how close will that guide cut to my ideal cut site. So for really knockout and knock-in experiments, we recommend testing multiple guides to see which one gives you the highest efficiency. For knock-in experiments specifically, it's very important to test out a few guides, figure out which one of those guides gives you the highest level of editing, because when we try to add in our donor sequence, only a portion of the edited cells will take in that donor. So it's a secondary cut that takes place. Well, cut's a bad word. Only a portion of those edited cells will take in the donor, so it's a percentage of our edited cells will have that insert. So it's very important to start at the highest level of editing that we can so that the end result is we have the most amount of cells that have that insert of interest. So those are a few ways to design guides. There, as we mentioned early in the talk, there's two critical reagents necessary for conducting gene editing experiments. The first is the guide RNA, which targets our region of interest, and that's what's going to target where we're going to cut. And then the Cas9 nuclease, and that is the piece that will actually do the cutting. So when we're thinking of reagents, unfortunately, there's not just one format that exists. So there's a few questions that we need to ask ourselves when we're doing the planning for our editing experiments. The first is, for my specific cell type, what methods can I use to deliver the reagents? There's a few key ones that we usually look at, the first being electroporation. So this would be kind of electrocuting the cells and then bringing in those reagents. If we're doing that, we're likely going to be using what's called a ribonucleoprotein. So that's a complex of both a synthetic guide RNA bound to our Cas9 protein. The other option is to do lipid-based transfection. Many different reagents can get packaged into there, so we can use synthetic guides and various nucleases, so we can use protein or mRNA there if we wanted to do that. Or the other option is to do a transduction. So we can load up reagents into viral particles, drop those viral particles onto our cells, and use the viral machinery to inject the sequences into those cells to be integrated into the genome. It just comes down to what methods the cells can withstand. There are some cells that do not like to be electroporated, so that leaves using a lipid-based transfection or transducing in our reagent. So, that's the first critical question, is what's in bounds, what's out of bounds for the cells of interest. And then the second question is, what is the downstream analysis that we need to do? If we're doing knock-in work, there are some formats that we likely would not use. For that example, I would say for knock-ins, we want to edit the cell, put our donor in, and then have that be stable. If we were to use something like lentiviral Cas9 particles coupled with lentiviral single-guide particles, what would happen there is, because they're viral-based, we would have integration of both sequences into the cellular genome, and those would constantly be being expressed, and editing would be always on. So if editing is always taking place, we can edit the cell. If we were trying to do knock-in, we would knock in a donor, but then editing would still continue to take place in and around that donor, making it very difficult to analyze downstream. So if we're doing knock-in work, we're likely going to be looking at the transient side of editing, so maybe using a protein or an mRNA or working in a stable cell line that expresses Cas9, and then just injecting in our guide RNAs, our synthetic guide RNAs. Synthetic guide RNAs eventually do get degraded within cells, and once they are degraded, the editing stops, even if we have a stable Cas9 expressing cell line, because you need both components necessary to make the edit. So there's a few options there. On the transient side, we've talked a little bit already about synthetic guides coupled with protein or mRNA. If you have a cell line that has Cas9 expression, whether it's stable or inducible, you can also use synthetic guides there. And all three of those options will give you quite a transient, a short-lived editing experiment. In the middle, though, you have an option where you can have an inducible Cas9 cell line. So this is an integrated Cas9 into the cellular genome, but it only gives you Cas9 expression when it is induced. So Horizon has an option for this, that the Cas9 gets expressed only when doxycycline is present on top of the cells. So what you can do there is, even if you're adding in, even if your cells don't work well with electroporation and lipid transfection is not working, so maybe you have to use lentiviral, you can use a lentiviral single guide particle with a lentiviral inducible cell line and not have to worry about that continuous editing that compared to if you were doing both a standard always-on Cas9 lentiviral option with the lentiviral single guides. So it gives you some flexibility in when you want expression versus when you don't. So the length of editing is really dependent on how long the drug is on the cells. So many options here, and it kind of comes down to what type of cells are we working with and what is that downstream analysis. So now we've selected our reagents, we've designed our guides, all of the pre-work is done. And now we're going to move to the bench and actually conduct the experiment. The first thing I would check is to make sure that our delivery is optimized. We talked on the last slide, there's three key methods. We can do electroporation, lipid-based transfection, or viral transduction. I don't have information on here about electroporation, but if you're doing electroporation, you'll likely have to test a few different of the protocols to see which one works the best to get your reagents in the cell, at the highest likelihood of getting reagents in your cell. The graph that I have on the left is lipid-based transfection, and what you're focusing on there from an optimization perspective is what is the cell density and how much lipid-based transfection reagent are you using. So for the graph on the left, we kept the cell density constant, and we varied our lipofection reagent. what you'll see is you get to a point where the cell viability drops. So what ends up happening is you do increase your editing efficiency with the more lipofection reagent that you use. That makes sense because you get reagents into the cell. But there's a point of diminishing return where you can actually have a drop in cell viability even though your editing efficiency is increased. So what this graph shows is once we get up to that 0.13 micrograms per well of the lipofection reagent, we start to see a drop anywhere from like 20 to 25% of cell viability. So at that point, we probably want to back off and use the 0.07 microgram per well. And this will all be cell dependent. So just mess around and see where that drop takes place. If we're using viral particles, though, so we're transducing our reagents into the cell, there's a few different things that we need to be aware of. The first is we likely want to have an MOI of around 0.3, and this means we'll have the odds of having 0 or 1 copies of that viral payload in each of the cells. We're not going to have multiple versions of that in each cell, and that's important. But the next thing to look at is we want to make sure that we're choosing a promoter within that viral backbone that has a high activity within our cell line of choice. There are not hard and fast rules for this. It comes down to testing different promoters in the cell lines. And one of the most interesting things that I had run across is the data that we see here in the middle is from a human cell line. And what I would think is that a human promoter would give me the most expression. And in this case, we see similar expression of both a human CMV as well as mouse EF1 alpha. So this just proves that we need to test multiple promoters in our cell lines and not just assume that just because we're working in a human line, a human promoter is going to give us the best expression. So what this shows is if we look at the promoters that give us the most expression, those end up giving us the highest editing efficiency, and that makes sense. The more Cas9 expression that we have in the cell will likely give us a higher editing efficiency, and that's what we see on the T71 gel on the right. So the highest editing efficiency of 18% exists in both the human CMV as well as the mouse EF1 alpha, which are the two leftmost images that we see there. And the last thing that we'll likely want to do on the bench is look at enrichment. So there are inherently some limitations depending on the types of cells that you're working with. Maybe there's few good guide RNA designs that exist within the region that you're trying to edit. So what are some ways around this? If you don't want to, what are some ways around to enriching these cells to where we can get our answer at the end? There are a couple primary ones that we look at. One way is just brute force. So you can screen more clones in order to find that rare editing event. This can be done if you have automation in your lab. You don't need automation, but it'll be more laborious because you're screening more clones. You'll have to start with more cells. It's just inherently more laborious. The second option, though, is to use a selection strategy. So maybe we have fluorescently labeled Cas9, a fluorescent reporter adjacent to our Cas9, or there's antibiotic selections that we can make use of, and this can help quite dramatically. So the data that we have here is a lentiviral Cas9 that has a fluorescent reporter adjacent to it. So with this, what ends up happening here is whatever cells bring in Cas9 will be fluorescent. And if we compare the unsorted to even the low concentration sorted, low FACS concentration, we see around a 3 to 4 times editing efficiency. So just by doing this one-step enrichment, you can dramatically reduce the amount of clones that you'll have to sequence on the back end if if you have to sequence them. So there's a few different ways to look at that, and that's a one step. You can even take it a step further, and if you're using a lentiviral guide that maybe has antibiotic resistance or a different fluorophore, you can use a secondary sort where now you've sorted out the cells that have Cas9 in them and that have your guide RNA in them. So maybe this editing efficiency increases further, or there's just a much more concentrated pool that you'll have to do your downstream analysis with. So there's many options here, and they can dramatically have an effect on how much work you're going to be doing in your next step of actually doing your downstream analysis. So now you've done the experiment. The question is, how do we analyze this? So I've broken this graph up into two halves. The top half is phenotype. The bottom is genotype. So that comes down to what do I need to know. And then the x-axis really looks at throughput. So on the left, low throughput. On the right, we have quite high throughput options. So I'll start on the bottom. If we're asking a genotype question, so maybe we're knocking in a tag or we're trying to create a SNP, we want to know the genotype of that cell line. So at the lowest throughput option, we don't really get great genotype information. This is the mismatch detection assay, so this is like a T71 assay. We use this a lot internally to spot check for editing efficiency. So you won't really get specific sequence information, but it's a nice quick check to see if editing actually took place. If you move a little bit further right, though, you move into the sequencing realm. So Sanger sequencing or next-gen sequencing, both of these would be required to figure out what is that new sequence that exists in your cell of interest. And for this, the decision really would just come down to how many clones do I need to sequence to push you one way or another, Sanger or next-gen sequencing. On the phenotype side, we may want to start by looking at a Western blot. Now, this doesn't give us actual knockout information, but I always look at this as a leading indicator. If I'm not seeing a band on my Western blot for that protein of interest, there's a high likelihood, but not necessary, that my protein has been knocked out. One of the reasons why it may not show up on a Western, but you may still have function, is if you edited a region that your antibody would bind to, and now that antibody can't bind, but the rest of the protein is functional. So it's a good spot check, similar to T71, but it's not going to give you full functionality information, but it's a good start. The next two things we can look at from a phenotype perspective would be phenotypic screens. These would be like drug interactions, like what happens when we hit these cells with a drug of interest. This can range anywhere from low throughput to very high throughput, depending on how these are arrayed out, how many wells are present. And then on the other, a little bit higher, potentially higher throughput, looking at FACS sorting. So one of the things you can think of here is if, say you're targeting a cell surface receptor on a cell, and you want to knock that out. You can have a fluorescent antibody that would traditionally bind to that cell surface receptor, but if you knock out that protein that gives you the receptor, your antibody won't bind, so you can do a negative sort with FACS sorting to pull out those cells that have had the knockout occur. So this, once again, this just comes down to what was my initial question that I had when I set out to do the editing experiment, and marrying that with the technique that's going to give me that answer. And that really brings us to the end of kind of the editing workflow and all the considerations that we should be aware of when we're setting out to do editing experiments. If you still have questions, I'd really recommend anybody on the call to check out Horizon's website. We have really great information on selection guides for reagents. So if you want a little more insight into when you would select one format over the other, please, there's great work there that can help guide you with that. And then once that selection's been made, that we have workflow diagrams for each reagent category. So step by step, what is necessary if I use a synthetic guide coupled with mRNA, or synthetic guide bound to protein versus using an inducible Cas9 line or even lentiviral options. So step-by-step guides regardless of the reagent format. And then we have application notes. So we get a lot of questions around editing in primary cells. So how would we go about doing this in iPSCs, T cells, or doing in vivo work? We have some application notes and posters that can give guidance around there. And if you're doing knock-in experiments, there's a few other considerations that I didn't get into a lot of detail in with this presentation. Things around how do you go about designing that donor, what format should the donor take, should that donor be, and things like that. And we have additionally some posters and application notes that can give guidance for knock-in experiments. And throughout the process, if there's questions that you just need a little more help with, we have a really great technical support team. They're specific to our CRISPR portfolio, available by phone, chat, or email. Whichever is easier, they're there to help. So, at the end of the day, I hope this presentation was beneficial for those of you, regardless of where you are in the process. If you're just thinking about bringing gene editing in, or if you've done it already, just want to hope that this can give some background on the step by step, what things should we be thinking about as we're planning, conducting, and analyzing our data for gene editing experiments. And with that, I just want to thank everybody for listening.